my name is Yazmin and I'm a junior here at the University of Arizona, and this summer I'm working in Dr. Pablo Pires' lab in the Department of Physiology.
Hi, my name is Viktoras. I am a freshman here at the University of Arizona and will be majoring in physiology. I am currently studying with Dr. Pires' lab, who is also in the Department of Physiology.
The main objective of Dr. Pires' lab is understanding the blood flow of brain in disease states. The current animal models we're using are known as 5X FAD, also known as having the familial Alzheimer's disease. This summer, I was able to help on the project that targets the TRPV4 gene and the use of vasoactive compounds such as GSK and HD606. We harvested the superficial and deep cervical lymphatic vessels in the 5X FAD and wild-type mice. By doing this, we're able to use these same vessels in what is known as a technique as pressure myography. By doing pressure myography, we're able to create a graph that compares the lumen diameter of time in comparison to the length of the experiment to be able to track the vasoconstrictions and vasodilations of these vessels. In this clip, we have a successfully cannulated superficial cervical lymphatic vessel. During this experiment, we treated the vessel with 3 nanomolar GSK proclaimed TRPV4 activator. As you can see, it constricts and dilates per the physiological environment we stimulated. At the end of the project, I was able to have a couple questions that continue to linger on. These questions include, how can the use of these vasoactive compounds translate to clinical care in regards to lymphatic complications? A second question I have lingering is, are the results of these vasoconstriction and vasodilations applicable to research of other body systems and organs?
In the lab, we use various genetically modified mice. This summer, my project was to genotype these mutations for preceding experiments. For genotyping, there are 5 main steps. In the first step, we use DNA extraction. Then we do PCR, then gel electrophoresis. Afterwards, we cut out the gel with the DNA, do a DNA extraction out of the gel. Once we extract the DNA from the gel, we send it off for sequencing in another lab. During DNA extraction, we use various detergents, buffers, and incubation periods to isolate raw DNA from unnecessary matrices. PCR is necessary for copying billions of DNA sequences. There are 3 main steps: denaturation, annealing, and extension. Denaturation separates the double helix into single-stranded DNA. The annealing stage occurs when primers bind to target sequences to distinguish which sequences to amplify. Extension occurs when polymerase creates new DNA copies from the existing base pairs. Extension stage doubles the amount of copied DNA and amplifies targeted DNA sequences. Soon after, we prepare an agarose gel, then load the extracted DNA for a process called gel electrophoresis. Gel electrophoresis uses electrical current to pull slight negatively charged DNA molecules toward the positive electrode through the agarose pores. The pores help indicate genetic presence based on the molecular size of DNA. DNA gel extraction consists of physically cutting out the indicated gel band. The gel extraction also utilizes detergents, buffers, and centrifugal techniques to separate the gel from the raw DNA. Throughout the summer, I have experienced some lingering questions. Without DNA, how would cells generate unique characteristics with their structure and function? If lymphatic vessels in the brain have an increased permeability, can amyloid beta proteins leak through the vessel?
Thank you for watching our video!